Hello everyone, my name is Avinash Kumar and we are discussing about the Streptococcus. This topic is included in the course code BMLS3001 for the course name Systemic Bacteriology, and this is the course is run in the School of Medical and Allied Sciences in the program BSc Hemolytic. So prerequisite or recapitulation before the starting of the slide is Streptococci are gram-positive, non-motile, non-spore-forming, catalase negative cocci that occur in pairs or chains. Older culture may lose their gram-positive culture. This is the recapitulation or prerequisite for this slide. Now about the objective of the slide, to learn about the Streptococcus bacteria. The objective for this slide is to learn about the Streptococcus bacteria. So now introduction, what is the Streptococci? Streptococci are gram-positive cocci arranged in chains of varying length and in pairs. Streptococcus form the part of the normal human and animal flora. Streptococci, here you see the Streptococci. This strepto means in the chain, branches form. Strepto means in the chain and in the branches form. Here cocci means the shape of the bacteria will be circular. So if we are seeing that we have Streptococcus, it will be look like this. Now it is said that Streptococci are the gram-positive. It means the cell layer, cell wall of the Streptococci are made up of peptidoglycan. Peptidoglycan layers are the thick layers as compared to the gram-negative. The basic difference between gram-positive and gram-negative are their cell wall, because in gram-positive the cell wall is made up of peptidoglycan, whereas in the gram-negative bacteria the cell wall is made up of bilipid layer, which is arranged in a chains of varying length. There will be a variation in the length, and in pairs. Streptococcus form the part of the normal human and animal flora. It means the human and animal flora are the bacteria which are present in our body, which are either beneficial or non-harmful for our body. Now the classification for the Streptococcus are on the basis of hemolytic classification. If we are doing the classification on the basis of hemolysis, there are three types of hemolysis. One is alpha hemolysis, second is beta hemolysis, third is gamma hemolysis. And the bacteria is known as alpha hemolytic bacteria, beta hemolytic bacteria, and gamma hemolytic bacteria. Now the morphology, they are gram-positive spherical cocci. They are gram-positive spherical cocci. The size is 0.51 micron diameter. This is the 0.5 to 1 micron diameter. Long chain. It is arranged in long chains due to successive cell division. Occurring in one plane means it is occurring in one plane like this. And daughter cells are failing to separate. Non-motile, non-sporing. That is non-motile and non-sporing. Non-motile means it will be not movable. It will not move anywhere. It is non-motile. Non-sporing means they will not generate different daughter cells. Now culture characteristics. What are the culture characteristics of the Streptococcus? These are aerobic and facultative anaerobes. Aerobic means this will grow in the presence of oxygen, and facultative anaerobes means if there is some lack of oxygen or lower concentration of oxygen, they will grow. Optimum temperature for the growth of the bacterial culture is 37 degrees Celsius. Now, which type of culture media is required for the Streptococcus? Blood agar. Blood agar is required for the culture media. Colonies are small, 0.1 to 1 mm in diameter after 24 hours. So the 24 hours is the incubation period. It will look like a semi-transparent, low convex, show beta hemolysis and diffusion 2 into medium. Mucoid colonies in any capsulated strain. Capsulated strain means if there is a cell, there will be some capsulated cell wall. This is capsulated strain. Now lab diagnosis. Lab diagnosis for the Streptococcus. Specimen which we are using for the collection of bacterial culture or bacterial contamination. Collected depending upon the nature of lesions. Collected depending upon the nature of lesion as pus from suppurative lesion. Pus from suppurative lesion, swab from sore throat infection, blood from septicemia, CSF from meningitis. Here septicemia means infection in the blood, meningitis means infection in the CSF. Infection or inflammation in the CSF. What is the full form of CSF? CSF is cerebrospinal fluid. Cerebrospinal fluid. Now moving to further, we will make smear and we will do ground staining. When we are doing ground staining, we can observe that the pus and other specimens show gram positive spherical or oval cocci arranged in chain or pair. So this is the reference and this is done for the day. Other bacteria we will discuss in the next slides. Thank you very much.